Gayatri, can you unscreen this? Unscreen the Gayatri?
Sir, okay, sir.
Sir, shall I start, sir?
Yeah, we can go ahead, Dr. Sanjeev.
Respected Director, NRRI, Madam Padmini Swami, Director IARR, Dr. R.M. Sundaram, Speaker of the Day, Dr. M. Seshasai Sriman, Professor, Plant Physiology, University of Agricultural Sciences, Bengaluru, Secretary of Society for Advancement of Rice Research, Dr. R. Mahendra Kumar, Secretary of Association of Rice Research Workers, Dr. Baik, other dignitaries attending this event, retired rice researchers, staff and students of NRRI and IARR, all the equipped cooperators and members of SAR, members of ARRW, and ladies and gentlemen, good afternoon to all of you. As a part of Azad Ka Amrit Mahotsav, the 75th year celebration of independence, on the behalf of organizing committee, myself, Dr. Sanjeev Rao, scientist biochemistry IARR, welcome you all for this 10th webinar jointly organized by Indian Institute of Rice Research in association with Society for Advancement of Rice Research, Hyderabad, and National Rice Research Institute, Qatar. The agenda of today's program includes opening remarks by Director NRRI, Madam Padmini Swami, followed by introductory remarks by Director IARR, Dr. R.M. Sundaram, introduction of today's guest speaker by Dr. P. Raghuveer Rao, Principal Scientist, Plant Physiology, Indian Institute of Rice Research, talk on developing rice cultivars for water-saving cultivation, a physiological perspective by Dr. M. Seshasai Sriman, Professor, Plant Physiology, US, Bengaluru, comments by Director IARR, a brief question and answer session wherein members can either raise your hand and wait for your turn or post your questions in short sentences in the chat box, which will be attended sequentially. Dr. Akshay, Scientist Plant Physiology, and Dr. K. Shruti, Scientist Hybrid Rice of IIRR, will be acting as reporters for this program. Closing remarks will be given by Director NRRI and also Director IIRR. It is followed by virtual felicitation of the speaker. Vote of thanks will be proposed by Dr. R. Mahendra Kumar, Secretary of SAR. This Zoom link supports up to 500 participants, and this program is available in YouTube. There is a general request, please keep your lines muted until and unless required. With this, now I invite Director NRRI, Madam Padmini Swami, to give her opening remarks.
Thank you, Dr. Sanjeev. A very good afternoon to all. Respected Director, Indian Institute of Rice Research, Hyderabad, Dr. R.M. Sundaram. Today's distinguished speaker, the veteran plant physiologist, Dr. M.S. Sesasai, Professor Plant Physiology, US, Bangalore, all the head of the divisions and scientist colleagues from Indian Institute of Rice Research, Hyderabad, all scientist colleagues from NRRI, Cuttack, all dignitaries and scientists joined in the virtual board, all participants, students, researchers, ladies and gentlemen. I really congratulate Dr. Sundaram for organizing such an important webinar today and inviting me to this webinar. This is organized in the background of Azadi Ka Amrit Mahotsav, as Dr. Sanjeev has already told, that celebration of the country as we are completing 75 years of independence. In this connection, many organizations all over the country are organizing different activities, and as a part of this, today's webinar is also organized. As we all know, rice is the world's most important cereal crop, which is being grown about 7,000 years or before. It grows in top hills of Jammu Kashmir to below sea level in Kerala, rainfed upland to deep waterlogging conditions where other crops are cannot even survive. In that condition also it is being grown. It feeds 65% of the population in India and about 56% population globally. 90% of global rice is produced and consumed in Asia, and it consumes about 80% of total irrigated freshwater resources around the world. Being a crop of largest water footprint, cultivation of rice in conventional transplanting method utilizes approximately 5,000 liters of water to produce 1 kg of rice. But nowadays, many water saving technologies we have developed to minimize the utilization of such a huge amount of water for producing same equal amount of rice. So water for agricultural use becomes increasingly scarce due to the climate change and rapid industrialization and urbanization. By the year 2025, irrigated rice of 15 to 20 million hectares in Asia will suffer water scarcity. As we know, rice production is important for global food security, but given its large water footprint, efficient irrigation management strategies need to be developed. Presently, some water saving technologies and practices are being followed by researchers and also by farmers, like alternate wetting and drying, aerobic rice, direct seeded rice, SRI methodology, micro irrigation systems, etc., for judicious use of water and for sustainable rice production. Along with all water saving technologies, it is also very important to develop rice varieties which are befitting for such type of growing methods. While developing any varieties, it is important to know the physiological mechanism or processes for which the plant is becoming tolerant or susceptible. And after identifying that particular important trait, it should be incorporated in the process of development by any means, all means of developmental methodologies like traditional breeding methodologies, genetic engineering, molecular, biotechnological means, even nowadays very burning CRISPR-Cas technology by knocking out the genes which are not desirable. So in this connection, today very befitting webinar lecture Dr. Sundaram has organized, and we are having with us the very brilliant physiologist with us, who is Dr. M.S. Sasai, who is going to deliver a lecture on developing rice cultivar for water saving cultivation, a physiological perspective. He has done excellent work in his career on plant physiology, many aspects of plant physiology, and I had opportunity to work with him many times to have discussion, and in N22 mutant project we are partners, and many times I have visited. Really, I am fortunate. I learned that root structure, what he has developed in, that is his indigenous methodology. He has developed in his institute how to study the entire root of rice when we are growing under aerobic, means under abatic stress like drought condition. So many things we have learned from him, and he is a veteran scientist of particularly that carbon isotope C13, C12. I do not know today's topic, it may come, or definitely it will be coming, because water use efficiency he is going to tell. So that special facility they are having in US Bangalore, and he is the master of that particular facility. He has done a lot of physiological work on abatic stress, particularly on drought stress tolerance mechanisms, and I think he definitely will have a very strong insight from his lecture. And not only we all scientists, our breeder friends also, those who are developing crop varieties, they will definitely gain a lot. I wish we will have a very brilliant and fruitful lecture today from Dr. Sesasai. As I told, he is very brilliant physiologist, and maybe in physiology perspectives, he is going to give us many insight, and particularly for judicious use of less water to grow more crop from the minimum amount of water. I wish a very successful lecture as well as a very learning lecture from Dr. Seshasai. And I again congratulate and thank the organizer, Dr. Sundaram, and his team for organizing such a nice webinar. And I think we should not spend more time. Better let us, anyway, Dr. Raghuveer is going to introduce our speaker. He is having a lot of very good career, and again we will hear to Dr. Seshasai. Thank you, Dr. Sanjeev.
Thank you very much, madam, for your valuable opening remarks. Now I invite Director IARR, Dr. R.M. Sundaram sir, for his opening remarks.
Thank you very much, Dr. Sanjeev. Respected Dr. Padmini Sven, Director NRRI, my colleagues from Indian Institute of Rice Research, Dr. Mahindra Kumar, Dr. Sanjeev, Dr. Raghuveer Rao, and all my other colleagues from All India Coordinated Rice Improvement Project, colleagues from Sister Institute, and all the members of audience. I extend a warm welcome on behalf of Indian Institute of Rice Research and also National Rice Research Institute, and also on behalf of Society for Advancement of Rice Research and Association of Rice Research Workers for this important webinar, which is jointly organized by both IARR and NRRI on an important topic, developing rice cultivars for water-saving cultivation, a physiologist perspective, to be delivered by Dr. Seshasai Sriman, who's serving as professor in the Department of Plant Physiology at University of Agricultural Sciences, Bangalore. I would like to qualify Dr. Seshasai Sriman as a veteran physiologist, but who is still very young at heart and also in his wit. And he has been a sort of mentor for me, and we have worked closely in a couple of committees, and I've been interacting with him. And let me take this opportunity to inform all of you that this year Dr. Seshasai has been identified as an esteemed member crop production for our institute also. So let's give a big hand to him, and then we'll be keeping interacting more, at least for 3 years or more. And Dr. Seshasai is also a special invitee for our annual rice workshop, which will be held in the 2nd or 3rd week of April. You're welcome, sir. Welcome to the rice family and also to the All India Coordinated Rice Improvement family. As Madam mentioned, like say, water is life. Similarly, rice is also life. We need to see the ways and means to grow rice in less water and then maximize our outputs. This is equally relevant because of late we have been seeing that a lot of rice is being exported out of the country, particularly non-basmati rices, which are generally fall under the bracket high volume, low value produce. So there is also a counter thought telling that by exporting, say, about 13 or 14 million tons of non-basmati rice, we are exporting almost 60 or 70 billion tons of water. So we are all working towards saving or minimizing application of water, both in upland rice and also in irrigated rice, in all forms of rice cultivation. In this regard, we have several varietal solutions and also solutions through development of hybrids which can save water. I would not like to enumerate them, but one critical component which we all should consider is the crop production aspects, wherein a team of agronomists, soil scientists, they are working towards developing water saving technologies which can minimize the application of water. Equally important are the two basic sciences, that is physiology and biochemistry, which will help the breeders or the biotechnologists, or rather the varietal developers, to understand how we can save water through various physiological and other mechanisms. And as much as breeding is important or genetic studies are important, I feel that basic studies in physiology are the fundamental for breeding for water saving conditions in rice. And in this regard, I'm very happy that Dr. Sesha Dr. Seshana has agreed to deliver a talk on an important topic covering his vast experience in this particular field. And I once again welcome you, Anna, for this particular meeting. And we are just eagerly waiting to listen from you. And we are also ready to bombard you with a lot of questions. There are many areas wherein we don't understand, or our understanding is very minimal. So pardon us if we ask any silly or naive questions, but we'll be happy to listen to you, not only during the lecture and even beyond also. Thank you very much. Sanjay, we can go ahead.
Thank you, sir, for your valuable remarks. Now the platform is set for the presentation by the speaker. Now I request Dr. P. Raghuveer Rao Garu, Principal Scientist Physiology, to introduce the guest speaker.
Good afternoon, everyone. I take immense pleasure in saying a few words about Dr. Sheshasai Sriman. Dr. Sheshasai Sriman has done his MSc, BSc from US, ICAR-IIRR Bangalore, and technologist. Working through for his lifetime, he has been working on water saving technologies. Particularly, he has been awarded. He joined as assistant professor in US, Bangalore, and thereafter he became a professor in 2010, and he's continuing as a professor from 2010 onwards. And some of the honors and awards which have been conferred on him are: he is the first prize in state level seminar on biotechnology in 1986, Best Young Scientist paper presentation in 1994, Outstanding Research Performance Certificate by DST in 1998, Boycott postdoctoral fellowship. He visited Farquhar Laboratory in Australia, and is member of Review Committee for Genetic Manipulation, member of Expert Committee for Young Scientists in SERP, and he elected member of the National Academy of Sciences, Allahabad, INSA Best Teacher Award in 2016, and elected fellow of the National Academy of Sciences, Allahabad 2018, special invited to PS SERP, and is a member of DST, DBT, and several other committees, and is also a privileged member of the RAC of Indian Institute of Seed Science and Indian Institute of Rice Research. So far, he has published 136 publications, and several of his publications are well noted and well cited regularly. And the major achievements of Dr. Sheshasai Sriman are: he has established a national facility for stable isotope studies and standardized several robust, high throughput phenotyping approaches to assess genetic variability in water efficiency, transpiration, and nitrogen efficiency in crop plants. Sir, he has released one such genotype, KMP 175 Daksha, has been released for commercial cultivation in Zone 6 of Karnataka state and notified as a cultivar suitable for semi-irrigated aerobic cultivation at the national level. And this is an honor for any physiologist developing a variety and releasing it for cultivation in Karnataka. And this laboratory has been contributing enormously for the aerobic rice cultivation and water saving technologies in rice. And towards this direction, he has discovered QTL regions governing variability in water efficiency, fruit traits, and other drought adaptive traits by association mapping, and validated the markers QTL. Sir, he has also worked on genetic enhancement of and bioavailability of nutrients in ragi. Not only his work is restricted to rice, but he works on ragi also, and that has also led to a good development. So development and deployment of SSR markers for mulberry linkage and QTL analysis has also been done by Dr. Sriman Sheshasai. The last, sir, I would like to emphasize that calculation of rice mutants for functional genomics has been taken up in a big way by Dr. Sheshasai, and he has been doing a wonderful work at this level at the Institute of very laureate from this institute have come up. He was a student of Dr. M. Uday Kumar and Dr. He has developed transferred a technology, sir, the rice cultivar named KF175, developed specifically for semi-arid aerobic cultivation, has been released for farmers cultivation in the southern dry zone of Karnataka. So these are some of the laurels of Dr. Sheshasai, and I take immense pleasure in inviting him for this talk today. And he has published 136 publications in various reputed international and national journals. He has visited several countries and participated in seminars and webinars. And today, sir, I am very fortunate to talk about him as he is my senior colleague. And I also congratulate him that tomorrow is his birthday, 20th February is his birthday. And I want to congratulate him in advance. So thank you very much, sir. This is a brief introduction of Dr. Sheshasai.
Thank you very, very much. I think I am already tensed. Dr. Padmini and Sundaram, I think you summarized what I had to tell. And thank you very much, Raghuveer. I shouldn't have sent you the biodata at all. You read everything. I mean, it's something like it makes me feel very humble. Um, it's something that is you are expecting, I think, a lot from me, and that's why I'm very, very tensed about it just now. So, is this screen visible, sir?
Yes, sir.
Yes, sir. Yes, sir.
You have to
Yeah, I'm putting it into the
presentation form.
No, I think it should be bigger and visible. Okay, so thank you very, very much. I think it is an honor for me to be a part of the RISE family, as you very rightly said. And as both the directors of NRRI and IIRR said, I have known and worked with both Dr. Padmini and Meenakshi Sundaram for at least a decade or maybe more. I think my beginning of the entire RISE research or the RISE journey for all this actually started in Cuttack. I was a young assistant professor when I went there, and I remember Padmini used to help me, and Dr. Patra, whom I don't know if he is there in the audience, I really thank him because one day when I was there, when I was there was some kind of a strike by some tribals, and we had locked ourselves into the germplasm unit. We collected the germplasm, and then that's where it started. That was way back in, I think, 2000, no, 9, maybe 2001 or 2 or something like that, quite some time back, maybe 20 years ago. That led to the whole program of developing the so-called rice cultivars for a water saving situation. There is something that I really want to warn you. There are, I'm sure, scientists from genetics, physiology, and biochemistry, and everyone. There is one thing that I will warn you now. Physiologists are very famous for confusing you. and and there are two things that I always tell: either you confuse others or you try to convince others. Let's see if I can sort of confuse you and at the end convince you that what is the way that we are doing is okay or whatever it is. So what we do is, I mean, this is something that we all know. I mean, thanks to the breeding activity that is being going on in India, excellent. We are happy, and I believe there was a news a few days ago saying that the world. I mean, this year has been the highest production, in terms of grain, food grains of 340 million tons. That's fantastic. But yet, the problem still remains is India is ranked among the lowest in the hunger index. And I'm not putting the nutrition index here. The malnutrition is very, very high. And though there has been an improvement, you can still see, people who are malnourished, children who are underweight or stunted, wasted and stunted are some medical terms that they use, are still alarmingly high in India, though there is a big decline in child mortality. And that's the reason why the longevity is sort of increasing. So despite all these progress, we are still poor in our food production as well as nutrition. That's where the securities are. The question that we always have: can plant physiology help at all? Even if it is a little bit, let us see if we can help each other. So, okay. And this is some kind of a data that was published last year, about a year ago, exactly. There they talk about how the water utilization pattern is happening in India. In 2011, there was a lot of water here. But then what's going to happen now is rest of the places are pretty safe. But then critically, a lot of places in India are going to be deprived of water. The kind of groundwater utilization is extremely high, and we are pumping the water resources out of the globe. I mean, that will be very alarming indeed. The water is going to be the most important crisis for food production. But the question is, so what? I mean, we always ask this question. If somebody says water is the problem, I say, so what? Get water and do something. But we know that the moment rice, though Dr. Padmini did say it has grown everywhere in India, from the Himalayas till the below sea level kind of situations. But there is a lot of water that it consumes. We know this, and this particular crop is quite sensitive to water stress. There was a big program that we wanted to compare, though I mean, for want of time, I didn't want to use that, though I used only this slide from that work where one of my students, Preeti, she worked in association Wageningen University at Netherlands, and where we compared how rice and wheat differ for adaptations to drought. But I'm not telling that story. But then when you look at the rice story alone, you have a yield loss that happens when the stress occurs at any time, whether it is the vegetative stage called the G1, GS1 stage, or the anthesis period, flowering period, the GS2, or the grain filling period. In all these cases, the yield loss is because of several other reasons, several specific reasons, such as the reduction in the leaf area or the canopy cover if the stress occurs early, and then the spikeless sterility, and then the grain filling inability are, I think, the most important reasons what causes yield loss. So if the GS3 stress occurs, the yield loss is very, very high, causing both spikelet sterility as well as grain filling. So we need to understand the why I'm saying that is we all know this, as a fraternity who has been working on rice for a long time. You are the champions of these things. You understand this very, very well. But the question that I have is, how is it that we can mitigate this situation at all? So there are only two things. Both the directors said this. How do we cope with this? You do agronomy, manage the water resources, manage the soil, and then you do genetic enhancement by breeding. Now, what the hell is a plant physiologist doing here at all? I mean, he can either watch everything or join the gang and then start contributing. Now, the question is, how do we contribute is the question that I have at least. So if the goal is to meet the requirement of the growing population in terms of food and nutrition, we need to look at two very important things. How capable are these plants to capture the resources, which is what actually happened during the Green Revolution time, where the resources were provided and the plants were made to capture the resources. But if there is a need, if there is a possibility that we can have another Green Revolution, I think it has to happen where the resource use efficiency becomes important. Now, when you put these two together, you have two situations. One, you have the resource unlimited condition or resource sufficient condition. We need to increase the yield potentials, or we call it the potential yield. And then under conditions when the resources are limiting, whatever may be the resource like radiation, or nutrition, or water, that is a situation where we talk about how to reach or how to enhance the realized yields under the given conditions. So we need to increase both, and that's where the challenge is. Really, we will have to look at now how we can address this. One of the best ways, or one of the ways that has been actually followed all through, has been breeding for selecting higher yield. We call it as absolute yield or yield per se. But long number of literature, in published literature suggests that it may not be the correct strategy in the future, for the reason that among the high yielding varieties, the yield has already reached a plateau, and we know that there is a ceiling. And yield-based selection may not be the one that should be used. And then there is a high genetic to environment variation. The interactions are very high for yield. I mean, that is true for every tribe, but for yield, it is because so many things actually contribute to the yield ultimately. Therefore, further breeding requires a very different effort from all of us. So I generally wanted to do this. There are two choices that we have at this stage. One is you identify a drought-tolerant genotype. That is what is being done conventionally, or it is called the empirical selection strategy. And the other one that we would like to do as a physiologist would be to look at drought-tolerant trait. I mean, there is a reason why we wanted to argue on this. This is an analytical selection strategy, and we need to analyze. I remember this when I joined as a young assistant professor. My senior colleagues, my teachers in genetics and breeding, they say, what is the use of plant physiology? We will only select yield. It doesn't matter. I still remember Dr. Vidya Chandra who used to tell me this. I don't require any of your costly equipment. I need my palm. I'll go hold the panicles in my palm. Whichever occupies maximum space in my hand is the high yielding variety. Oh, wow, isn't it? I mean, if that is as simple as this, now we wanted to look at if there are 10 varieties which will occupy almost the same space in the palm, we do not know which one you use it for crossing programs for further development. The reason is the drought tolerant genotypes that we pick up, the one that occupies maximum space in your hand, may not have the other good qualities. Maybe it is tolerance, its levels of the disease resistance or whatever other kind of thing, taste, cooking qualities, whatever may have differences. Instead, it is something like what we want to do is you introduce the important traits into one, like analytically, so that the same variety that I want, which has all the good things in it, I will improve its productivity is one other strategy. Now the question would be, there are for sure genotypes which have been screened. All of you have done that. I remember Padmini saying that she has screened about 10 or 20,000 accessions of germplasm at CRRI Cuttack, NRRI Cuttack, by empirical ways. Now, if you look at the trait-based ways, we may say, okay, it is there, but then we ask a question, are such traits there at all? If at all they are there, what are they? There are these analyses that are done in the literature. This is one of those very early paper that was written by Matthew Reynolds and Roberto Tuberosa. I'm sure Matthew Reynolds is a physiologist from CIMIT, and Tuberosa is a geneticist and a plant breeder. He is in Italy. And you look at this, one person is in Mexico, the other person is in Italy, and they write a fantastic review article quite some time back. It's almost like 12 years ago. So what they do is they talk about different traits that we look at. And then again, the whole thing depends on the famous ICAR-IIRR yield model, where we say yield is water used, the transpirational water used, and the efficiency with which that water is used times the harvest index, partitioning, will determine what is the yield. It's a simple, very, very simple, straightforward model that has been used by a lot of people, including us. We made a little better analysis of the whole thing, and we said, if you wanted to have a drought adaptation mechanisms, we need to have two major things: maintain water relations and maintain carbon metabolism. So you maintain the two things that would take care of a lot of things. In turn, the maintenance of carbon metabolism and water relations depend on several traits, like we classified them as morphological traits, cellular level traits, and then we have, most importantly, the physiological traits that are integral traits. Now we have we can go on and on into this next round of complications. This is the reason why I said physiologists will confuse you. If you were to be a breeder, you will just look at drought adaptation by growing them under water limited conditions, pick up the fellow that grows the best, simple. But then if, what am I saying? Morphological trait, change a lot of them, then physiological trait, tell most of them. I mean, I would have put 10 more here, but for want of space and wanted to make it look sort of one, what is that, symmetrical structure, I removed so much of complication, but it can be still complex. But if you want me to simplify the whole thing, it just goes towards only two traits. Anything that you do to maintain water relations, anything that you do to maintain carbon or any metabolic activities would definitely lead to adaptation under water limited conditions. That's the simple thing. So our concept has been that drought tolerance can be improved by bringing together these many diverse traits inside. Now, how to do that is what I will discuss in the next 25 minutes. Improve the adaptation, there is no doubt about it. And then we try to do that in many different species, compared between wheat and rice, compared between, let's say, habitually plant kind of species like groundnut, sunflower. In all these cases, what we wanted to do was to improve the adaptation. When you look at rice, to semi-irrigated aerobic condition is the major target for us. And then if you can save water by at least something like 60%. Yeah.
Hello, sir. Sir, slides are not moving, sir.
Slides are not moving?
Yes, sir. Here we are not seeing.
It is moving here, no? It's for me.
Sir, you can do one thing. You can unshare and then once again try to share from the slide which is presented.
Sure.
Sorry to disturb.
Oh, there is a resume share. Is it? Can you see that now?
Yes, sir.
Yes, sir.
Yeah, it is moving, right?
Yes, sir.
Yeah, something happened there. It said resume sharing. I must have pressed something. Right. Okay. So this is our concept where we are trying to save water, but then without reducing the yield potential. This is where the challenge is in terms of combining them. Okay, so when we look at the scientific strategy for this, we wanted to identify the relevant drought adaptive mechanisms or traits. And then, we all know how to go ahead in the next step is to identify the genes or QTLs and then adapt molecular, molecular breeding or any of the omics assisted breeding mechanisms and put the traits together. That's all. Basically, what I'm trying to do is you're trying to tell you one step, a little step before already whatever that you're doing. If we can add this, perhaps we can do things better. As I said sometime back, there are two traits: maintain metabolism and maintenance of water relations are the two most important things. And we calculate, we can simply classify them as maintenance of tissue water relations depend on root system. Maintenance of carbon metabolism depends on photosynthesis, which determines water use efficiency. That's all. But I generally, when I tell this to students, I keep telling that suppose you go for a war, the war that we are waging is against drought or water limited condition. Productivity is what we need. And you go to the war, and then the soldier goes which well equipped, like he knows how to use a gun, dagger, sword, bow and arrow, everything. But he is not brave. On the other hand, there's a very brave person who just wants to go there and fight the war because he is very patriotic. But both these guys will die. This guy who is equipped, he is not brave, he will never go to the war field. The other person, very brave, not trained, will go and die straight away. So this is not the way we should fight the war. Therefore, what we hypothesized was the maintenance of tissue water relations and the maintenance of the carbon assimilatory or the water use efficiency mechanisms can happen only when the plants have a much higher propensity to respond to stress. And that we call as a group of traits, which I will tell you about a little while later, is called the acquired tolerance traits. These are not the traits which are present normally under when the plants don't experience any stress, but they acquire that suddenly when the moment the plants experience stress. It will be something very similar to the priming kind of thing that we practice. Now, if you go through this particular story about the root has been done by a large number of people. One of my physiology colleagues who is in Baramati, Dr. Jagdish Rane, was a part of this paper in Nature, which came in 2017 about the importance of root. And then they tried to now find out how to now have a, they identified the DRO1, allele, deep root in rice mechanism. And then they showed IR64 can actually become like this and therefore become very highly tolerant to drought. What we did was to do a very similar kind of an exercise here. We analyzed two sets of them. Dr. Padmini was talking about the mutant resources that we had generated in one of the projects that we screened a large number of them at very many places in India. And the mutants as well as a large germplasm, which I said I took it from Dr. Patra from CRRI. And then of course, now we have a whole lot of these 3K panel genome. Seshu Madhav, who was there from ICAR-IIRR, is also a part of one of our national fund projects. So what we are trying to do here is we have the root structures. Dr. Padmini was also talking about that, and we can very easily screen the root variabilities. And when we now see this, we identified some mutants which have high root, low root, and then we identify what is the mechanism in which they have. The high root ones will always have better photosynthesis or better metabolism under water limiting conditions. This was published by one of my PhD students in physiology of plantarum recently. But there's a problem. I mean, there's a long story that I'm cutting short. We can discuss about that when we meet, in detail a little while later. But we know that there is a variability in the root architecture, root system architecture, or root traits, length, the angles, and so many things can be measured. But we know if the deep rootedness exists, there's a possibility that the crop can exhaust the water from the deeper layers of the soil more easily or quickly. So that has been very well recognized internationally, and we also, did some review on all those things. And therefore, we need to look at what the other traits are. If you are putting only the roots, they are definitely possible that they will enter into a late season stress, or the stress will become a little more severe in the, let us say, later phases of the phases of the plant, such as the GS2 or the GS3 phase. The yield loss will become much larger if the deep rootedness alone exists. Therefore, we thought that why not we increase the root, ask it to go access water, but use it very efficiently so that the biomass increases. So the question was, how do we capture the genetic variability for the traits that we're looking for, which are tough? And again, Dr. Padmini and Sundaram did say this. And then we work a lot on the stable isotopes. To cut the long story very short, we know this has been a very well worked out phenomenon. A lot of people across the globe use this technology called the carbon isotope discrimination or carbon isotope fractionation method for identifying genetic variability in water use efficiency. And the theory of why and how delta 13C, we call it as our carbon isotope discrimination, is associated with water use efficiency is extremely well worked out. So let's not go into the details of those things. And what we can see here is in a large number of species, especially rice, we have done that in a very, very large number of accession. And we showed, I mean, this was a hardcore measurement of water use efficiency by gravimetric approach, really. And that was associated with water use efficiency. That was quite some time back in 2005, we published it in Crop Science. And that led to a whole lot of work in carbon isotope ratios. And of course, this was originally started by this gentleman, Professor Graham Farquhar, and this guy who was there, he visited, both of them visited my lab quite some time back, Professor Richard Richards from CSIRO, and he is Graham Farquhar, with whom I did my postdoc in Australia, in Australian National University. They were the ones who started the whole program on water use efficiency by measuring the carbon isotope ratios. And they were the first to actually release the varieties called Brisdale and Rees in Australian very dry conditions, in the Western Australian conditions. I mean, it was really nice to go there and see those kind of trees. So we also wanted to do similar kind of things in India. We established a national facility which consists of the isotope ratio mass spectrometer. And this is from a company called Thermo Fisher Scientific. And for the past 20 years, we have been working tooth and nail together. And it's a lovely exercise to do this. I mean, some of you must have visited. I'm sure Padmini has been here and Sundaram had been here. I would really welcome anyone to please come and use these facilities. It's a national facility. But again, I'm very good in confusing you. Now, what I'm trying to say is, I said, if I have roots only, then the roots will exhaust the water and therefore it will get into stress. But if I increase water use efficiency, again, I'll confuse you. Now, water use efficient genotypes will perform only when there is stress. If there is no stress, if there is season rainfall is high, then the grain yield advantage decreases. Wow. So that means increasing water use efficiency is not a good one. Increasing the root is also not a good one, but both of them are important drought adaptive traits. Now, what am I saying? What I'm saying is, when we looked at the germplasm, this was the rice germplasm that we got from Cuttack. This was about 200 germplasm we saw, but we classified them based on root. And we did a similar kind of things where the groundnut germplasm was, which we got it from ICAR-IIRR, are also compared. Now, what happens is these are low root genotypes, and these are the high root genotypes. And in these genotypes, if you see here, carbon isotope ratio is high in high root genotypes. Now, if you see this graph here, you'll see that if the carbon isotope ratio is high, water use efficiency is low. This is what I wanted to remember. Now, when you get back into this, once again, the high delta 13C suggests that the water use efficiency is low in the high root genotypes. Now, this is the problem. Now, people like Abraham Blum from Israel, and he was one of the strongest opponent of breeding for water use efficiency. He would say it is effective use of water, or the EUW rather than WUE is what he used to say, like reverse it. But the differences between our understanding and his philosophy was that the low root genotype has high water use efficiency. But if you pick the high water use efficiency genotype among the low rooted fellows, genotypes, then you are most definitely to reduce the biomass. Low root genotypes have low biomass, low yield, and it's so true in germplasms of rice as well as in groundnut. We say the physiology remains more or less the same, independent of these crops, but the determination of these physiological traits to the growth, the productivity remains the same. That, therefore, when you picked up for high water use efficiency, because they're experiencing stress, low root fellows, water use efficiency increases, but that resulted in poor growth. This is the problem. So what we did was we again picked up these germplasm, we compared them for similar leaf area. If you have no differences in the leaf area, which is causing a lot of variations in the total biomass, then carbon isotope discrimination and biomass were strongly related, suggesting that if I take out the variability which is caused because of other major traits which are integral to the crop, then efficiency parameters will start becoming important for determining the productivity. Same thing that happened even when you had similar or comparable transpirations. At a given transpiration, differences in water use efficiency definitely determines the differences in biomass and yield. Therefore, we say that genotypes with high water use efficiency, they will not show, they will have high total biomass if the other variabilities are taken care. This is where the concept was. So how to now make measurements on, let us say, the transpiration was the question. This again took us back to the stable isotopes, and we know the theory about how water gets enriched with heavy isotope of water during evaporation of water from the oceans, and we translated the ocean story to a simple leaf, and lo and behold, it was an extremely beautiful discovery that we made in our place. This was, I think, one of the very early reports of how oxygen isotope can be used as a surrogate for measuring transpiration rate. And after that, we improved that, and then we said at a given leaf area, carbon oxygen isotope ratios or oxygen isotope enrichment that we talk about can be a good estimate for root rates. So we combined the two stable isotope strategies as phenotyping mechanisms, and then we wanted to look for both the isotopes, finding out differences in root as well as differences in water use efficiency. So though we have some kind of a variation that the high root genotypes have low water use efficiency because they transpire more, it is also possible that there are genotypes where the high root genotypes may have high water use efficiency, and those genotypes must produce more biomass. There's no doubt about it. Therefore, we wanted to analyze further what are the subcomponents of water use efficiency that we can bring. This is one of the programs that I mentioned about Seshu Madhav, who was also there with us in one of the NASF funded projects. What we are trying to do is to now break this water use efficiency further down into subcomponents. And then there are two physiological processes: photosynthesis, which is called the net assimilation rate here, and the stomatal mechanisms, which are called the mean transpiration rate, are the two parameters that strongly contribute to differences in water use efficiency. And we further divided that into saying that the mean transpiration rate depends on the stomatal number, frequency, and the size and the extent of their opening, which we call all of them put together in plant physiology terms as stomatal conductances. And then the net assimilation depends on how many tissues are there, how much of volume of cells are there in the leaf, which is determined by the cell specific leaf weight, or in some literature they say LMA. LMA is leaf mass area, which determines the total surface available for biochemical processes of photosynthesis. This is just a cartoon to show this. If the leaves are thick, we measured the leaf thickness in about 150 germplasms of rice, which is a part of the 3K panel for which we have the whole genome sequencing. And then we measure the stomatal frequencies, the leaf weight in terms of SLW, the leaf thickness, and then we identify the contrast. I mean, this doesn't mean because there is tremendous variability in so many traits that we look at, but we wanted to now pick up genotypes which had differences in these parameters. So what we did was we grew them, selected about 32 genotypes which represented the variability that we have in the stomatal frequency and the leaf thickness by a specific leaf weight, and we measured the contribution of these things to the net assimilation rate. See, basically what we are trying to do is we know that the water use efficiency will improve biomass, and we break that down into NAR, net assimilation rate, and mean transpiration rate. And then we are trying to now find out if specific leaf weight it can contribute to the differences in net assimilation rate, there to water use efficiency, then to biomass. So that's the kind of regression that we need to do. So what we observed was, when you look at the specific leaf weight, there was absolutely no relationship with net assimilation rate under well-watered conditions. It is 100% field capacity. That means there was no need. There was no, even when there is so much of available resources, why do you want anybody to be efficient? It's general, simple thing. If you are rich, you will never be able to spend anything. But the same thing happened in a different way when the stress was there. When the moment the plants were grown at 60% field capacity, the entire thing aligned, and therefore there was a much stronger relationship between net assimilation and the specific leaf weight. But it turned out a very beautiful other trend when you looked at the stomatal frequency and net assimilation rate. Stomatal frequency contributed to the photosynthetic rate under well-watered conditions. You can keep the stomata open. You can make many stomata, no problem. There is no dearth of water to transfer. But if you have many stomata, actually the metasimulation that does not depend on that because they have to close there, or it can be detrimental. Therefore, the conclusion from here or the confusion from here is we must have genotypes which will have more number of stomata and those leaves which are thicker. Only those perhaps will do better under water limiting conditions, the aerobic growth, which is what I will show you in this. So we have the water use efficiency of the thin leaf and the thick leaves. Thick leaves are more, where definitely under the well-watered, water limiting conditions. Thick leaves are, they're actually not very good one under well-watered conditions. And then you have the few stomata and the many stomata kind of situation. And this also shows a very clear trend where if you have many stomata, the transpiration will be more and the water use efficiency will be less when compared to the few stomata business. Now, what happens to the water use efficiency if you say all of them put together? You can see here, few stomata and thicker leaves will have the highest water use efficiency, and the many stomata and the thick leaves will have the highest biomass. Therefore, the effect of thickness and the stomatal mechanisms may override one another, but they will adjust in such a way that we can help them to put more biomass. What we wanted to do was, this much of physiology was fine, but then we wanted to check and find out if this entire thing can be translated into a program with the breeding mechanisms. So I thank my breeding colleagues, especially Dr. Rajana and Deepak, who are here in our university. What we did was we identified, again, with the germplasm collection that we had actually got from CRRI, Cuttack. I call it a CRRI, it's NRRI. And then we crossed that with one of the genotype which has high water use efficiency with a high root genotype. And then we developed a recombinant inbred line. There are all these stories that we have measured the water use efficiency in the several seasons. We identified the consistent ones and the differences in the water use efficiency. Root was different. You can see that the IET 15963 had the highest root, whereas this other genotype that we talk about, Tanu, had high water use efficiency because of low delta 13. So we crossed them. We developed recombinant inbred lines. We thoroughly phenotyped them and identified a specific line. A number of those lines which we identified, one was called the KMP 175. KMP is Karnataka Mandya Paddy. Mandya is a research station where we do this. So that's the name they have given in the research station. Is perfect. Preeti, one of my PhD students, worked on, when she was doing her master's, she did this experiment where she did a whole lot of analysis of the physiological mechanism that was improved in that KMP 175. Again, the same thing, the root length was high, the leaf area was high in KMP 175, carbon isotope ratio was lower. Though it was not lower, it was not better than water use efficiency of Tanu, which is the water use efficiency donor. It was not as good as that, but definitely it was better. You can see that the biomass and then the yield. Yield generally I don't give in this because the criticism is, we should have unit area yield. I should have picked up that slide. Now, this particular variety, which gives more biomass, has about 26% more yield under water limited conditions than any other checks that we had. And I was extremely glad. I was actually excited. This is the only thing that I have done so far where we released a variety for farmer cultivation and we named that variety as Daksha. Daksha in Sanskritam is efficient. So this Daksha has also been nominated, and Dr. Rajana here, who's my colleague breeder, helped us to develop this variety. He has sent it already for PPV FRA notification of this variety. Like I said, to go into the next step, we said that these traits are very good, but then what is most important seems to be is how good are we in terms of making this entire thing brave and face the entire thing. Therefore, we require what is called the acquired tolerance. Acquired tolerance, as I said, is something that does not exist under well-watered condition, but they are significantly upregulated specifically when the plants experience stress. So we used to do this simple thing called the temperature induction response, a priming kind of a thing with using a very, very simple growth chamber. which can be controlled in terms of its temperature and damping and all that. And we have been working on this. Professor Uday Kumar has been driving us to do all these things. One thing seriously I miss is my Professor Uday Kumar. Anyway, what we did was to show that when you have the propensity to respond, or the cellular level tolerance, and we showed that when cellular level tolerance or the acquired tolerance is high, and combined with that of, let us say, deep-rooted mechanism, they will have the highest productivity in terms of, because here talk about the percent reduction in spikelet fertility. You have the high root biomass with high acquired tolerance. You have the lowest reduction when compared to low root biomass and low CLT. I mean, it's cellular level tolerance, or it is acquired tolerance. When the acquired tolerance is low, you see that the spikelet fertility is very, very low, or the percent reduction in spikelet fertility is very high. Okay. What we did was to develop a platform. So to do these things, not at in these experiments, we had to do it only at the seedling level, maybe 2-day-old seedling, very highly heterotrophic kind of situations. If you wanted to do it in autotrophic condition, Lakshmi did this experiment with us. What she did was to grow these plants in a platform that we have created. Please, you're welcome to visit us one of these days. Maybe your workshop that you're planning, we should have it in Bangalore. And you can visit this facility. We have about 900 millilisimeters. Each one is controlled specifically to give a specific water treatment to that. Water regime can be maintained independent of differences in the root or canopy or the amount of water that they transmit. It's all automated here. So we can actually ramp the speed at which the soil is dried in the container to mimic what happens exactly in the field conditions. And with that, Lakshmi did this experiment to now understand initially how we can quantify the acquired tolerance traits. We did that initially with 2-3 genotypes, 5204, BPT 5204, which is a very highly water-loving plant. I mean, a plant which is grown under irrigated condition, when compared to another germplasm line that we got from CRRI again, AC 39000. This particular germplasm line has very high propensity to respond to water-limited conditions, or the one that has higher level of acquired tolerance. And when we measured a large number of oxidative stress damage mechanisms, like the O2 minus production or the membrane damage that we have, and the total antioxidants capacity, you can see that the intrinsically tolerant germplasm had much better than BPT 5204. And we have also done that with IR64. IR64 is still worse than BPT. But please be aware, both BPT and IR64 are good yielders. There is no doubt under well-watered conditions or under puddle conditions. But the moment you put them to water stress, look at this membrane damage significantly increased, oxygen free radicals production significantly increased, membrane damage in terms of the melandialdehyde content, which is an indication of the peroxidation of the membrane lipids, increased significantly. Whereas it was not like that in the case of AC39000. You can see very low here, and all that. Suggesting that these antioxidant capacity correlate tremendously with that of the spikelet fertility parameters, and therefore it correlates very well to the yield. You can see that the spikelet fertility determines the yield to an extent of 87%. These are R values, but R square will be close to 90% of this. Depends on the spikelet fertility. It is obvious. And the spikelet fertility in turn depends on how good we are in terms of managing the oxidative stress. What we wanted to do was, this is what I said already, that those rings came now. And this is another, again, it came again to say that this, this is not pending, it was accepted already. I don't know why it is pending, it is printed. What we did again was to now take these two genotypes of rice, another genotype that we added from the germplasm, which were all classified based on acquired tolerance and the root traits. The moment you have these two traits differences, you can see high acquired tolerance and high root had a spikelet fertility of 56%, whereas low acquired tolerance and the low root had only 28%. This is a very, very small spikelet fertility under the water limited conditions stress. You can see that under well-watered conditions, it could be different. Percent spikelet fertility is very high for these things under well-watered conditions. The decrease, of course, but then the reduction is something like very, very less. And you can see that the yield is already gone under stress, but this guy is maintained much better. The one that have high acquired tolerance. What we did was to do a metabolome analysis. This was done by one of our other PhD students called Pushpa. We worked with NCL Pune National Chemical Laboratories, where a non-targeted metabolome was done, and a large number of things came up. To cut the long story short, again, what we did was we got all these kind of things, and then we wanted to identify where these are. In the sense, we had done a QTL mapping of this, and then we had identified two markers sitting very close to each other. And these two markers were also associated when we used the whole germplasm line for making the measurements on all the acquired tolerance traits. And this, in this particular region, there were about 16 or 22 genes, which were all in the rice genome. But in that, there are 2-3 genes which were extremely beautiful, were the glutamate decarboxylase and then the raffinose synthase. GABA is something that maintains the membrane, and then the raffinose synthase is the one that maintains the water relations. Now, again, it adds value to whatever hypothesis that we made for testing, that it is water relations or the maintenance of metabolism that will actually is responsible for this. And this particular genes actually co-localize into the same place, which are flanked by two markers which are less than 1 centimark apart. So therefore, this is very, very nice. What we did was further on, this was done by two of my PhD students, Raju and then by Pratibha. They took this 175 indica line that I got from CRRI Cuttack, and that was phenotyped, and then we genotyped at that time with SSR markers, and we identified the QTL, validated them using biparental mapping populations, and developed the lines which are trait in progress. This is what I want to tell you. This is something that you all know. There are a lot of diversities in that, and then the population had population structures revealed, and there are 6 subgroups in it. And then we identified this is published in Molecular Breeding some time ago. And this was another case of doing the marker-assisted selection using the 3K panel genotypes for acquired tolerance. And these are the variability that we find in the germplasm for many of the oxidative traits. And then, we had about 300 SSR markers in the germplasm in the first step, where we studied the root and water use efficiency QTLs. But when we recently did this experiment, we had 240,000 SNPs, which had a mean allelic frequency of more than 0.5. And then that showed a very beautiful story. And then the trait diversity was quite large for many of the traits that we measured in those germplasm. And then we did, you look at the Manhattan plot to now identify QTL that are associated, markers that are associated with several of these traits. Once again, let's not get into the details, but to tell that there are a large number of markers that we can get for specific trait that we are looking for. And these markers can be used for trait introgression. What we ultimately proved was the genotype that had an advantage of high root, high water use efficiency, and high levels of acquired tolerance had very low reduction in speculate fertility. That means that speculate were very fertile, and that resulted in high yield. That was pretty simple in all the germplasm that we have tested. And what we did was we did a marker-assisted back-cross breeding with all the markers that we had developed for the traits that I mentioned. We took IR64 as that was one of the mega varieties that are grown and loves water, and we crossed it with this to trait donor genotype, 39020 for acquired tolerance and root, and then IET 16348 for water use efficiency. We combined the two F1s. We had a large number of SSR markers which worked for foreground selection, and we used about 120 markers spanning the whole chromosome, the whole genome of rice, at least 10 markers on each chromosome to work as background markers for reconstructing the background of IR64. What we did was, when we do the back crossing, took the F1, crossed it back with IR64 3 times, and then we selfed it. And then what we have now is BC3 F8 or something like that, and we have about 50 of these trait in progress lines which are actually doing very, very well. And here, when we can see some of the trait introgression lines, we compared for, again, all the physiological trait that we talk about. Leaf areas were better under water limited conditions, the trait introgression lines. The relative water content, or the water relations was better, water use efficiency, lower delta means higher water use efficiency, and speculate fertility was definitely better under the water limited conditions in trait introgression lines. But that's all fine. But if you look at this, yield had a big advantage. This is IR64, Under water limiting conditions, there's a huge reduction. The trait introgression lines maintains much better water use efficiency. And these lines, which have about more than 98% of the IR64 background, is performing really, really very well. And I should thank Pratibha. This was a very hard work by that girl who did this. Another girl who validated this in another set of populations. We took some 26 of them, and then we grew them in the phenomics facility and did a whole lot of physiological screening for these things and find out that the trait introgression lines indeed were introgressed in the traits. So that means the progeny that we have got now has high root, high water use efficiency, and they have high propensity to acquire tolerance. Once you have these things, they seem to ignore the kind of stress that they have. Now, this is the facility that we created. There was a video of about 1 minute, but somehow it is not running. I tried checking it out before I uploaded this set of slide deck. But this is a facility that each container is under the control of a software to add water, exactly how much water the plant loses. That means I can keep them always at the same level for all the plants. I mean, that is one of the most important things that we require for drought studies. So in summary, we can say that there is a clear understanding of the physiological basis for drought adaptation. And we can definitely work together with the plant breeding fraternity to now see if we can accelerate the whole process. And it was all the time the challenge was to phenotype these things under natural conditions, because most of the experiments that are done by the physiologist is done inside some controlled environment. You can see here everything is open, and therefore these are true to the kind of sense. But of course, you can say, hey, this cannot be true because you are growing them in containers. These containers that we have it can hold about 25 kgs of soil. So there's quite a bit of space for only one seedling can grow. You can look at the lush, how well, this is a germplasm line, so there will be all sorts of variabilities that we look. So phenotyping can be done under natural conditions. Now it can make a lot of sense. Genetic resources that we have in India is huge. I'm sure every one of you will have access to germplasm, mapping populations, mutants, and therefore, I think we are in the right time to now take a step ahead and just speed up and accelerate it. And therefore, with all the genomics effort that has gone behind the whole process, and we have the 3K panel with the whole genome sequencing done by IRI, and all these things can be now used for adopting this approach very strategically. And here is what I would always say is, we do the breeding, but for physiological traits. So why don't we call it as physiological breeding? And this was, I should thank really the funding that we have been receiving from DBT, ICAR, and Department of Science and Technology, CRB, and others. And I should thank all my collaborators. We have Ravi and I work in TNAU. I'm in the National Institute of Plant Biotechnology, like Anita, Rohini, Prashant, and IARI. We have Vishwanath and A.K. Singh, and Madan Paul, and a number of them. And of course, NRRI, we have Dr. Padmini Sen, who's here, Meera. They were all associated with us very, very strongly in the mutant program. And we have recently started this program in NASF with Seshu. And then Sarla, Dr. Sarla was there in IIOR, DOR, and Lakshmi Prayaga, who was physiologist. And we did a lot of work together again in the mutant program. And of course, we have a number of international collaborations with Wageningen University, Netherlands, and the ETH in Switzerland, then Kansas State University. Krishna Jagdish, I'm sure many of you know him. He was an alumni of our university, but he's doing very, very well in the United States now. And then, of course, in ANU, Richard Richards and Ram Farquhar. And I would be, it will be a mistake for me if I don't say thank you very much for these set of lovely people that work with me. They hate me, I know. But then at the end of the day, they are a happy lot. They laugh a lot. Thank you very much. I'll be very happy to take any questions.
Thank you, sir, for your excellent presentation. And IIRR appreciates your efforts to culminating 20 years of research in 1 hour class. And in your introduction, you said that you will be confusing as a physiologist sometimes, but the development of Daksha and other RILs that you have developed clearly indicates that you have identified convincing traits to develop water use efficient rice varieties. Thank you, sir. Thank you very much for your presentation. And you have covered from naked eye analyzable markers to machine dependent markers, and your metabolomics and all are also very convincing, and it's very inspiring, sir. As a biochemist, I can say that it's a very inspiring lecture. And with these words, I invite Director IIRR, Dr. R.M. Sundaram, for his comments on the lecture.
Thank you, Seshana. Your talk was indeed sort of eye-opening for me in many respects, because there were certain aspects like, say, we breeders or neo-molecular breeders, we work blindly. We're talking about single locus or multiple locus. We rarely, like, say, take into consideration the physiological aspects. I believe, like, while physiology is important, I think in the next phase of your career, there is physiology of disease and pest resistance. That aspect also, I think we can certainly collaborate. And we are working on certain pathosystems with respect to major rice pathogens. And also we are working with certain insect rice interactions also. I think it will be a win-win situation for IIRR, US Bangalore, and also for NRRA and other partners to work together in the area of stress physiology, particularly with regard to biotic stresses. Of course, in today's talk, you opened up an important facet related to another stress, that is abiotic stress, particularly water stress. It was indeed invigorating. I'm now 100% sure that Dr. Uday Kumar's legacy will continue, and then it will flourish beyond his dreams. And then we are all happy that you are continuing forward the excellent work that was done by him. And congratulations to you, and the best wishes for the group also. Before we open up the forum for questioning, Sanjeev, can I take the liberty of asking two or three questions to Dr. Seshana?
Yes,
Okay.
yes, sir. Yes, you're most welcome.
Yeah. In the era of rapidly changing climate, one of the most important traits which has been talked about, in addition to water use efficiency or efficiency use of water, is nitrogen use efficiency. According to your experience, what is the relationship between these two traits?
The relationship between water use efficiency and nitrogen use efficiency?
Yeah, I'm a little bit scared of using the term water use efficiency based on what you scientifically presented. Let's call it efficient use of water and nitrogen use efficiency.
Yeah. Efficient use of, efficient use of water and water use efficiency are same. But Abraham Blum says effective use of water.
Okay.
I mean, that is where the problem is. Effective use of water depends on the root only. And see, what I look at is that when you look at the nitrogen use efficiency or nutrient use efficiency, nitrogen generally, which should be used for making more of the metabolic activities, and if photosynthesis is an important determinant of water use efficiency, nitrogen use efficiency and these two together should go on. We did that exercise in groundnut quite some time back with the Australian scientists. So what we did was to now find out if we can calculate SLA. SLA is a surrogate for water use efficiency. And then we used SLN, specifically nitrogen, for measurement of nitrogen and all that. When you now put them together, there's a very strong correlation. So that means if a plant where the photosynthetic parameter or photosynthetic efficiency contributes to the efficient use of water in your term, and that fellow should also have better nitrogen use efficiency as well.
Actually, Dr. Padmini may need to go to join another forum. Madam, I request you to give your remarks about Seshana's presentation.
Yeah, excellent presentation, Dr. Seshasai. I congratulate you. And really, what Dr. Sundaram told, it is an eye-opener for many of us. Even if I am also in the same physiology line, but everybody has to learn from you. It is really excellent. And mostly the water, which is very badly necessary for growing rice, growing any crop, of course. And now we are going to face scarcity of that we are facing, and it is going to increase. And in that condition, how to understand your last slide, very clear understanding of the physiological basis of drought tolerance. That is really very, very important. We have to understand why it is tolerating. Like what you told that even if I have screened a large number of germplasm on empirical basis, only selecting genotypes. Also, of course, we have done some analytical basis also for some traits, so very few traits, but not so in-depth study like you for water use efficiency, stomatal frequency, carbon isotope discrimination, so many things coming to marker-assisted selection and metabolism, so many things you have told. It is really a good lecture, and I thought it will be never ending. You can speak along. I know that we had many times your lecture, but it is really excellent. And what you told that phenotyping is also very important on trait-based phenotyping is also very important for particularly for drought stress. And nowadays, how it is under natural condition we are able to do that also you expressed very nicely. And the genetic resources whatever are available, that germplasm you got from us or anywhere collected and did excellent work. Mapping population also, and mutant I worked with you also very nicely. You worked for giving all the physiological insight how to tolerate, and last the genomic based crop improvement approach, which need physiological breeding. That the terminology, I request all my breeder friends also, physiological breeding is very important, what Dr. Sesasai told, and we should have in mind that how to incorporate our physiological traits in the breeding program so that we can develop a very good, excellent, tolerant genotype, what is really particularly for water stress condition. So thank you very much, Dr. Sesasai. I think everybody might have enjoyed your lecture and might have also learned many things from your lecture. Thank you. Thank you, Dr. Sundaram, also giving me opportunity. I'm sorry, I will have another program in our institute, so I will just leave. Thank you very much.
Thank you very, very much. I'll tell you, it was very easy for me to do these kind of things for the reason that my first love in anything that we did when I was an undergraduate student was genetics. I mean, that was my first love, you know. I mean, I can never forget. Though I got married to physiology, my first love is always there behind my mind. So,
they're all interlinked, genetics and physiology, whatever you tell.
Actually, madam, yes, so actually, physiology and genetics people, if they work together, we can do real wonderful things. That is a very strong, because I mean, if you look at it very carefully, except you and Vishwa, I have all other colleagues that I have collaborators in India are all geneticists. So it's a very beautiful situation that we have. As Sundaram was saying, we have a win-win situation by putting hands together. I mean, I would very strongly suggest that we should start working with the breeders. I mean, they will not work with us. They are very, very busy. We have to go poke our nose inside and tell them, look here, boss, we can also do this. Please give us some time. I'm sure that can work out.
And also with plant protection specialists like
Oh, yeah. Sure. Yeah. I mean, it's we are all one, now everything is one Vasudhaiva Kutumbakam kind of situation. All things actually come towards physiology, whether it is insect or pathogen or drought or any abiotic stress. It's all mechanisms that help, therefore biochemistry and physiology should become important. I mean, we need to suggest that. Instead of that, what we generally do is, okay, there was this discussion that we had. What is physiology? Is measuring chlorophyll content physiology? I mean, that question we should ask ourselves. But thanks very much, Padmini Madam. I'm really, really happy. Actually, it all started here. I can see Dr. Patra also here. Patra. Actually, you
is there available.
You both started my journey. Thank you so much for that.
Thank you also. At least you remembered.
I always remember. Why? I have never forgotten, not even once.
Yeah. I may be allowed to, before our director leaves, may I interact? Dr. Sundaram.
Yes, sir. Go ahead, sir. Go ahead, sir.
Yeah, because
Right.
my director will be going away for another meeting. I take this opportunity to request our honorable speaker, today's speaker, Dr. Sasasai, whom we met maybe 20 years back, what he is recollecting. Maybe it will be more than that, probably before 2000, before the new millennium started, probably we met. So yeah, that's what I remember. But then, sir, whatever materials we had shared with you, I think all these materials have not seen the light of the day. If you provide the data to us, those materials can see the value. We can find the
Sure.
value in the materials.
Sure, definitely. I think I had sent once long back some email I had sent with all the data, but I will send it again. Absolutely not a problem. I will send you the entire thing.
Yeah, because what I am telling, tomorrow I'll be retiring, day after tomorrow you will be retiring. But the materials, if they see the value of this, the next generation people can utilize them, and the value, yeah, database also will be stronger, sir. Thank you very much.
Absolutely, sir.
For my request, sir.
And I will do that immediately.
Okay, sir. Thank you, sir.
Thank you very much, Padmini madam. Thank you very much, Dr. Patra.
Thank you.
I have a couple of other questions, but let's start with our opening batsman, Dr. Subbarao, our head principal scientist and head plant breeding, and also our head crop improvement, and also PA of breeding division in ICAR-IIRR. Sir, go ahead, sir.
Thank you, Sundaram. I've been waiting to talk to Sai. We have been associated. Actually, the association did not end in a fruitful way. I know Sai since late 90s, when he was for a brief period, he was at Mandya, and then I was handling a germplasm project. When the Datta was the DDG, he wanted me to address something which is below the ground. We are always talking about above the ground, always giving the data on agromorphological traits, especially in the yield and then some of the yield related traits. He wanted me very specifically to address something below the ground. And then that was the point I approached Sai to work on the root traits of the germplasm. And then it ended only at the discussion stage. I don't know the reasons why we could not go ahead, but now I want Sai to at least help us in doing that uncompleted, unfinished dream of the then DDG Sapandatta sir. And then I was very anxious to work with you at that time, Sai. Somehow you came once. I also met you once in Bangalore. We discussed. It was only at the discussion level. Never in the actually. It was not far. Now at least you are there with us. I think you will be there for another 2-3 years as a member of RAC. I remember. Is that right, Meenakshi?
Yes, sir. And in fact, I would suggest that let's have a plan for hijacking him like Veerappan, so that even beyond our annual workshop and REC meetings, we can retain him and then make him guide our youngsters and also the feeding program.
I was telling Vijayji should run to the breeders. It was the other way. I ran to you.
I know. I don't know why it did not happen. I remember now. If you tell me, I don't know why. Generally, it does not happen like that. If somebody says that, if geneticist comes and says, I want to work with you, I will be the first one to go do all the labor work. I honestly do that. I don't know why, what happened, Dr. Rao.
Meenakshi, you should support us now. At least we will do something which we wanted to do, which we could not do it.
Certainly, my pleasure, sir. We will certainly work together, Anna. And certainly, like say, there could be 1 or 2 topics related to physiological responses to pests and diseases. Also, something, it may be a new area for you, for us, but that's a very lucrative area wherein we can work together. In addition to working, of course, for water stress tolerance.
Absolutely. And then, one of the things that is coming up nowadays in a big way is the combinatorial stresses. What happens if there is a drought and the biotic stress like diseases and pests? Or what happens if there's heat and drought are combined or something else? There are all amazingly complex and easily workable things. We have the resources. I think we will only have to. I really am sorry to Dr. Subbarao. How did I do that? I shouldn't have done this.
We will do it. We'll do it, sir.
We will do it, of course. Yes, sir.
Because I have only 1.5 years of time only. And
like Dr. Patra said just now, it doesn't matter whether you and I stay. Somebody will do it, will create the situation. That's all.
Sanjeev, I think we can start with a regular rapid fire round for Dr. Seshana.
Yes. Yes, sir. Now I invite Dr. D.V.K. Nageswara Garu to ask his question because it's a very long text posted in chat box. I request Dr. D.V.K. and Rao Garu to interact with the
So
speaker.
you will have to unmute
Sir.
yourself.
So, hello everybody. In fact, I know you, Dr. Seshasai. I was in Rubber Research Institute of India for a long time.
Yes.
I know, in fact, entire physiology group of UAS Bangalore. Devakumar or Nanjareddy or even Nataraja Kadaba.
Yeah, James.
Including yourself, Dr. Prasad, Dr. Uday Kumar. Okay, anyway, I come to my question. In fact, I am a soil scientist. My strongest belief is the soil solution is the seat of all the chemical reactions at the soil root interfaces. And dilution theory definitely works. It should work, as I believe. But if you think about the soil plant atmospheric continuum, normally physiologists limit it to soil water plant relationships only. But for me, it is nothing but soil solution. If that is the case, what kind of attention is being given to the soil solution chemistry, at least within the plant system, and allied questions? I think basically this is my question, though I wrote in a lengthy way. So essentially, I say that soil solution chemistry is must, and dilution theory is to be regarded if you want to reap the best, because ion must be given a free hand to work, and that freedom comes only with dilution. So this is what I wanted to comment or know your reaction.
I mean, I have absolutely no doubt about it. Soil solution most definitely determines growth. There is no doubt about it. What we say, there are two things about the water use efficiency that we can look at. One is the agronomic way of defining water use efficiency, which depends on the soil solution, the soil type, texture, and everything. There, we add water and find out what is the growth and what is the yield and all that. But what a physiologist does is, after the water has been taken up, that is used in terms of transpiration, how much of that transpirational water loss actually triggers the growth rate of the plant inside the plant. But definitely you are correct, so correct that you can never ignore what is there in the soil. But when we look at the genetic diversity, we have to know, I mean, there are two types of people who can study physiology. One is you are trying to look at the genetic diversity within a particular species in a particular location. Other things are kept constant. What are the genetics of it? Number one. Another thing is, suppose if I want to understand everything about, let us say, some particular trait, then soil, plant, atmosphere, everything will come into the picture. So that is another thing. What we do is we screen the germplasm, look at the genetic diversity, pick up the best ones for crossing, take the contrasts for doing all these analysis. So we have contrasts for all of them. We need to sort of look at, I mean, this is exactly where we need to work together on all these things. And whatever you said is so perfectly correct. There is no doubt that the soil is the most important place where everything has to grow. People are, though they are talking about soil-less agriculture and all that kind of things, that is another story. But again, the chemistry of whatever that they do should be exactly the same as that what we have in the soil. We will have to work out on that, sir. But when we look at what genetic diversity is and how do we do crop improvement for that particular strategy, we look what, we generally want to do what I am trying to say. But if I try to understand how the plant behaves, then whatever you are saying is also important. There's no doubt about it. Two streams. And those two streams of research activity can actually have a crosstalk. There's absolutely not a problem.
Moreover, transplanted rice stands in the soil solution only, right?
Yes, yes. I mean, the soil solution, which improve, yeah, of course, it does.
So a footnote to Dr. Nageswara Rao's comments. It's not only physiologists and molecular biologists like me are confusing. Even soil scientists can also be confusing. But the confusion, through the confusion, they bring clarity also, which we got right now.
Yes, exactly. Basically, I always believe order comes from chaos. This is a theory in physics. And all the things were chaotic and then order comes. That is why sometimes I keep telling, let these politicians be as corrupt as they want to be, because order will come only from them.
Actually, sir, in 1971, there was one international conference exclusively on soil solution chemistry. Surprisingly, after that, nothing is heard about. I am really wondering, actually,
Why, sir? Why you did not talk about it further?
No, in fact, I worked when I was in rubber research, sir. We have developed certain centrifugal filters also. But somehow, I was moving to different institutions in the meanwhile. So there was a discontinuity. In fact, I thought of doing in rice for sure. And in fact, I started some work.
You can still do it.
We can
Exactly. I'm doing it.
Maybe when we all meet,
Very
we
much,
can do a lot of brainstorming on all these issues.
It's required, sir.
Certainly, sir. When you'll be here for about 3 or 4 days, you can stay for a day extra and then have a detailed discussion, sir.
Lovely, sir.
Yeah. Sanjeev, you can go ahead.
Thank you, sir. Thank you. Thank you.
Sanjeev, you are muted. Dr. Sanjeev, you are muted.
Next question is from Dr. Kalyani. Specifically, which traits should be focused while breeding for aerobic rice, as soil moisture needs to be maintained throughout?
What's that? Sorry, Sanjeev, what is that?
As soil moisture needs to be maintained throughout.
Her question is aerobic rice cultivation is a method of cultivation which is different from dryland rice.
Yes.
Wherein we maintain a minimum soil moisture throughout the crop growth period. So which are all the physiological traits which are required for essentially aerobic rice growth.
Okay, whatever I said today is all only for such kind of an environment.
Okay.
This is for aerobic cultivation. We call it a semi-irrigated aerobic. Is little less than less water than actually the aerobic, which is kept at 100% field capacity, or little more than that. It is less than saturation, though. There is no standing water. But if you want to really save 60% water, let us say you irrigate once in 8 days or 10 days, then first day you have a surface irrigation, quite a bit of water is there. But the third day onward, the soil moisture goes on depleting. And then depending on the condition of the weather conditions or the soil texture and all that, the rate at which the water loss can occur can be very different. This is what I said, that the rapid and the gradual stress that can happen in the field can be mimicked by the phenomics platform that we have. I think Sundaram and others, you should visit this platform, and then that will be very, very nice. Maybe in April, May or something, no, if you can.
Certainly, sir. When the cropping season starts, we'll be visiting.
Yes. We are doing the sowing now.
Yeah.
And April or sometime April, May.
We'll be there. Yeah.
At that time,
along with all the heads of sections, certainly.
Yep. That will be very nice. I'll organize that.
Sanjeev, before we move to the next question, Dr. Neeraja has raised her hand. Let's hear from her. And Neeraja, madam, I can go ahead.
Sir, good afternoon, everybody, and
wonderful,
Dr. Shesasai. So, as you might be aware, in the IIRR, physiology section is leading on nitrogen use efficiency studies, and with the support of NICRA and other projects now, Dr. Subramaniam, Dr. Buleti, Dr. Sanjeev, and of course Akshay, all of them are leading with support from soil science, and as well as our director was mentioning, we as a molecular breeders, we have pitched in. So as a member of our RAC and also as a use efficiency expert, whenever you are coming to Hyderabad, you should at least give us half a day time so that we can show you the facilities which we have developed under for nitrogen use efficiency studies, and also the material we have developed, and you can suggest our lead us or guide us the way we should go ahead. Thank you very much.
We'll certainly
accepted, madam. Actually, I know you. We have met before, and then one of the committees, and I have also visited the DRR when it was DRR, and I know Professor Virat very closely, and it's always a pleasure to work with an institute like DRR. It was always my dream, Sundaram. You remember we used to discuss that in Bara, whatever that some place got for second place. Balu Sahib, correct. And then I always wanted to be a part of you, and I'm very, very thankful to this this situation now. I will definitely, madam.
Oh, thank you. Thank you.
Sir, Dr. Ramesh Tatkunta from PJTSAU wants to ask you. Sir, Ramesh, sir, please go on.
Dr. Shesai, it was very heartening to listen to you. I would ask you two simple questions. One thing is how to quantify, means how much each trait would contribute. You have explained it by correlating with yield itself, but each trait, how much it contributes, means that is one. And second is, if you are not focusing on yield alone with the water use efficiency, what other traits you can concentrate? Because roots are most difficult, and we have done experiments, done projects, that's okay. But otherwise, above ground, something.
I mean, actually, Ramesh, you know what? Basically, what we are talking about is we have water use efficiency, we have root, and you dissect it further down because we only look at this way. My hypothesis is, if the hypothesis is wrong, everything is wrong. The hypothesis is, if a plant can maintain water relations in any given soil solution condition, if it can maintain water relations, that is, it is taking up water, keeps the cell turgid, and maintains the metabolism, that fellow should grow. That's all. It will be something like telling you should be intelligent and hardworking, you will be successful. Is there any doubt? There is no doubt at all. But
not like.
No, what I'm saying is, if you look at the above ground plant, we look at photosynthesis. I mean, that is very high, what you call as details of physiology is not necessary. What I mean to say is, I mean, that we do, we don't have to, tell that in some kind of seminar, like where everybody is there in a group. But what we do is we look at stomatal conductance, we look at the unproductive water loss, nocturnal transpiration, what proportion of that they have, what is the extent of limitations to photosynthesis, is it stomata that is contributing or the mesophyll conductance which is contributing, what happens to carboxylation efficiency. There are a million things that can be done, which are being done. But what I am saying is, how can plant physiology help crop improvement? Understanding, definitely, yes, whatever that you have to do, we must do it. But what is most important for us is crop improvement also. How is it that there are two things? One is crop improvement. How can I help as a plant physiologist? And I understand plant physiology as a scientist. What else can I do? There are different things.
No, simple extension of that question. I'm saying because roots are most difficult not to handle and things like that.
They are not difficult. Why do you say it is difficult?
No, not difficult for you. Not difficult for you, but
it is not difficult for anybody.
Anybody, okay, agreed.
I'll tell you why. See, now, of course, whatever that we are doing is very, very old, indigenous kind of thing that we are doing. And what Dr. Subara was also asking is similar. But now you have phenotyping. We have MRI scans. You can put the probe inside the soil and it will tell you where the roots are growing. I mean, nothing is impossible, Ramesh. That I know.
No,
agreed, of course.
You understand my perspective for a minute. That's all. So I'm not saying. So my perspective is roots are difficult. So I'm asking you, what other traits you can improve upon as a subject above ground, such that other discipline people can pick up easily. Maybe they can pick up roots also. I'm not denying on that. Most important.
Dr. Ramesh, pardon me for interrupting. It has been a grave mistake on part of all breeders and pseudo breeders like me to look at the plant parts and then ignoring the root component. That has been a gravest mistake because almost 90 or 100% of our irrigated rice varieties have robust plant type, but the root architecture is very, very poor in most of the varieties. And none of them even have some of the major QTLs which can allow them to tolerate. And as much as, of course, as much as root traits are important, Dr. Ramesh, I would also feel above plant parts are important, but there should be a mix and match somewhere wherein which I agree with your question. Like, for example, you say that in addition to roots, what else to look for? One or two. Is it the question?
Yeah, there's something like that. Something
the right way, Seshana?
Yeah. I mean, that is what I thought. There are many things that one can do. For example, we are also asking why. There is something that I didn't tell in this talk, but then we can discuss when we meet. Rice protein is most easily digested by human beings than pulse protein, if you know this. We have done some deuterium tracing experiment by human feeding. It's very clear that the rice protein is much better digested by human beings. But unfortunately, the rice that we eat has much less protein in it. Now, what physiology can be done to increase the rice grain protein content without decreasing the yield? That's the biggest challenge. And that will be fun. I'm sure it can be sort of cracked and we need to discuss about this. What other parameters one should look for? I mean, we need to have an enumeration of all those things, go on ticking and then removing them. I mean, it is, I cannot tell that, you do this, you do the other things and all that. We need to come out with a hypothesis, Ramesh, and that's exactly what I always feel. I mean, even for my PhD students, I will ask them, you tell me what you want to do. Now, let me say if it is doable or not.
Yeah, okay.
But if I tell you, there was one, I'll tell you why I don't want to say that. There was one person who was working PhD, was doing PhD in our department, it was Uday Kumar, and he had asked her to work with me. And she said, you write it here. What do you want? 3rd of the March 3rd, do it sowing. 20th, you do the weeding. I have to write like that. And she would go do the weeding on 20th, whether weeds are there or not. The weeds did come, she never went there because you did not write. So therefore, that is, I mean, I would say there are many things. Let us say pubescence on the leaf surface, or the leaf thickness, the thinness, the waxes on the leaf surface, cuticles. There are many things. I mean, this is what comes to my mind when you ask.
Just one interjection here. Dr. Sesha, at any point of time, have you worked with another cultivated species of rice, that is Oryza glaberrima?
No.
Because whatever traits you mentioned, larger and then larger stomata and stomata more in number, along with thick leaves, these are all hallmarks of glaberrima and then glaberrima derived mapping populations, which one of my colleagues, Dr. Girish, is working on. Fortunately, he's also a student of US Bangalore. So when you'll be visiting,
Girish, I know,
yeah. So when you'll be visiting here, he's exclusively working on developing rice varieties for direct seeded rice, but he has developed excellent population. If you require to screen some of those populations for those physiological traits, we'll be happy to share. And I think there'll be a wonderful reaction, Dr. Girish, if you can interact and then develop with Sesha. That'll be really
Yeah. I know Girish, and that definitely is.
Dr. Swami? You have to unmute yourself.
Sir, my question is very simple. It is easy to differentiate thin leaf and thick leaf. Similarly, is there any technique to differentiate high stomata leaf and low stomata leaf?
Sir, there are two ways of doing this. One is the typical way is go smear, take an impression, and go see in the microscope. And
I mean, in the field condition.
And the other thing that we are doing is to measure how much of water is being transpired. Now, there are other methods. For example, if you wanted a high throughput mechanism, if you have, let us say, a very large number of breeding lines, or if you have a very large number of germplasm to screen, we have sensors. For example, infrared imaging, and we have a camera which can now capture the infrared. And this infrared image that we now take will now tell you whether the leaves are cooler or warmer in a given environment. And that will be a very strong indicator of whether the stomatal conductances are different straight away. Pick up the contrast, get the number down to something workable, and then again do the conventional thing of taking the impression and then examine. I mean, if the examination is not possible in thousands of varieties which you have as a breeder, screen them initially and then pick them up.
Shall I read out the questions from chat box?
Yeah, go ahead.
Sumita Thomas is asking how to study drought stress on croplands.
How to study drought stress in croplands?
Yes.
I mean, you need to see there are many ways of doing that. One is you have to now find out what is the water content in the soil, like Dr. Rao was also mentioning some time ago. It's all whether you can calculate what is the texture, the structure of the soil, and find out what is the water content, and then calculate what is the field capacity. Then you know whether the soil is under drought or not. That's one way. That is the conventional way. But I would look at how does the drought stress response differ in plants, and that's what I am trying to do from one angle. There are many ways of doing. I remember Satish Verulkar, who works in a very high drought area in Raipur. He does it in a very different way than what we do. And there are many ways of doing the same thing. Not none of them are wrong, and none of them are, I cannot say this is the only way of doing things. I cannot say that. There are many ways. How to determine drought in croplands? I don't think that's a correct question. But I think the question would be, how does the plant differ in response to drought? I think is that the question that somebody is asking?
Yes, sir.
Okay.
Mika Thomas, are you there? Can you interact?
Just taking one question forward. I would like to know, Seshana, is there any differences in the physiological responses with regard to drought response at vegetative stage and drought response at reproductive stage?
Oh, yes. See, that's what we did first. That was Preeti's experiment in her PhD. What she did was to now grow these rice and the wheat plants in stress, give the stress at GS1, 2, and 3, vegetative, then flowering, and then the grain filling periods. When you look at that, the yield reduction was there, either the stress was at any stage. But the yield loss was highest when the stress occurred during the GS2 and the GS3 phases, which resulted in spikelet sterility. Whereas in the GS1, the reduction in the yield was mainly because of reduction in the leaf area. The canopy cover decreased, the leaf expansion decreased. I mean, that will again say Ramesh's question is what other above ground we can measure. Leaf expansion rate is something very sensitive. We measured that in the mutant population with Dr. Padmini was also associated. The leaf expansion of in rice, it takes about 12 to 14 days to get the full expansion from its emergence. And that is, it will reach its maximum length, maximum width. And if that gets affected, then the leaf area decreases. To that extent, biomass decreases. And so, there's a lot of variation for that.
Yeah, so that also in corollary means that it's tough to expect a variety which is tolerant at reproductive stage and also tolerant at vegetative stage. Or should I qualify?
Yeah, I mean, I can tell you that it is not very imprudent to think that there cannot be anything that exists. It can exist there. I'll tell you why. If you have better root mechanism to maintain water relations, the leaf expansion is taken care of. If you have acquired tolerance, spikelet sterility is taken care of. And if there is a mechanism which maintains high water use efficiency, metabolism is taken care of. Therefore, under stress, it can still make carbohydrates and send it for grain filling. It can be possible. What would be interesting? That's a good point that you said. I'm thinking about what you said now. We have the trait introgression lines, put them under different types of stresses and check what happens. If you have the trait introgress lines by breeding, you can create stress to them during vegetative, then flowering, then the grain filling periods, and then find out what is the extent of yield loss vis-a-vis something which is very sensitive.
That's exactly my question.
Fantastic. That will be an experiment which is very highly doable in our platform. We'll plan that.
I think I got it, Shishana. You can go ahead, sir.
Yes. Yeah. Dr. Satendra Kumar Mangrutia from IARR is asking what would be the ideal approach to maintain equilibrium of water and nutrients uptake and use efficiency?
What could be the
ideal approach to maintain equilibrium of water and nutrients uptake and use efficiency?
I still do not understand the question. What would be the ideal approach?
Or Satendra,
let me put in a right perspective. Actually, what I was, because from the presentation, what I could understand that reducing the root volume and vis-a-vis increasing water use efficiency is probably the ideal trait that we are looking for effective utilization of the water. Now, once we reduce the root volume, it will also adversely affect the nutrient uptake. So my question was, in this perspective, my question was, what would be the ideal approach so that we maintain equilibrium of water and also nutrient uptake and use efficiency?
Excellent. How are you?
Sir, I am good, sir.
See, Satendra, what I'm saying is, I don't know whether I communicated it or confused you as I always do, is small root genotypes will generally have high water use efficiency under stress, but their biomass will be low.
Okay.
Right? So therefore, we are not saying that the root should be small and the water use efficiency should be high. No. Root should be also big, but it should not lose water and exhaust it. It should use efficiently. That's what I mean. But when we need big root, long root and better root system, so that the nutrient uptake. And I think Dr. Rao was also asking about it some time ago. Nitrogen, Sundaram was asking, nitrogen use efficiency and water use efficiency must go together. Otherwise, if I don't have nitrogen, that means I don't have the proteins, enzymes, or metabolism, and I don't think I can ever have higher efficiency of metabolism. So, good root system is important for both nutrient and water uptake. There is no doubt about it.
Okay, sir. Agree, sir. Yeah. So, yeah, that's what my question was. And another question, sir, I had. So, since I am now there on the board, I can have another question also. Is there any connection between root hairs and leaf morphology? Because I was trying to understand that if the root hairs affect the leaf morphology, or is there any systematic study showing correlation of morphophysiological traits of below ground plant parts with above ground plant parts?
There may be something like that, but I'm not very sure about root hair and what determines the root hair and what determines the leaf and all that. I'm not very sure. But one thing I saw, there was a correlation when we had a mutant plant called a narrow leaf mutant in our rice. And this narrow leaf had two things. One was their roots were very thin and long, and the grain was also narrow and slender. Now, what is the connection that they have between in terms of the genetic determination? I'm not very sure. But there seems to be some pleiotropy here. But I don't know whether the narrow leaf, for example, there are genes which are associated with the narrow leaf, right? The NLM is a narrow leaf mutant. They have done that in both groundnut and rice, and I'm sure they would have done that in barley, wheat, and all that. When you have this narrow leaf, genes are mapped already, and what do they have to do to the root growth? I'm not very sure, but then people are saying that there could be some kind of pleiotropic influence of one another. Maybe because of the biomass that it produces. Narrow leaf will be narrow, less amount of carbohydrates made, and therefore there is not much of carbohydrates or like photosynthesis transferred to the root, and therefore the roots will be small. This is the kind of a corollary explanation one can give. But is there a cause-effect relation? That will be interesting to see.
Because I'll tell, sir, actually one of the study we were trying to overexpress one microRNA that affects the root hairs, and we were studying those root hairs, but incidentally we found that there are differences in the leaf morphology.
Absolutely.
So I was just I was like, is there any physiological connection between root
Yeah,
hairs?
possibly, because if these microRNAs that people are now talking everywhere, microRNAs, small non-coding RNAs, and all these kind of things are actually coming in the way of metabolism. And if they are inhibiting, if the microRNAs are responsible for root growth, the similar kind of thing should be happening even for the stem growth or the leaf growth. So if they are inhibiting the root, it will inhibit the leaf and vice versa, I'm sure. So there could be something, or there could be something which is common as well. We need to work out on that. I mean, root decreased because the leaf decreased. Now, how did the leaf decrease when root decreased by your control, your mechanism of determining smaller root? That will be an interesting story to investigate.
Great, sir. Thank you, sir. Thank you very much, sir.
Thank you, Chandra. Nice, you're there here.
Yes, sir.
Dr. Sridhar is asking for combining donors for crossing. Good combining donors for crossing.
Good combining donors of
crossing.
Yeah. I mean, that is obviously no. I mean, if the person who asks is a breeder, he will know better. What I will do is I can screen them. I will identify, let us say, 10 good fellows and 10 poor fellows and all that. Now, among these 10, who should be the one that crosses well? That we need to find out. I mean, this combining abilities, and then the kind of, my genetics friend Rajana who works with me and Deepak who work with me, they are always very happy to talk about what is called as broad sense heritability, narrow sense heritability, and the genetic advance over the mean, and all those kind of things are generally done, definitely, yes, to get to where we want to go. In the sense, suppose if I say I need to make BPT 5204 a drought adaptive variety instead of growing them under puddle conditions. That is one way. Another way is to create completely a new one. So if you are trying to replace the BPT 5204 with a new variety, that's a different game. Now, in these two mechanisms, that is, you improve the 5204 or IR64 or something, you definitely need to look at the specific combining abilities and then do the crossing. And we need to have that kind of data beforehand, I think. I mean, I'm not very sure about that technique anyway. All the breeders, Sundaram can tell me.
So I'm a pseudo breeder, rather. Okay, we can go ahead.
Yes, sir. The next question is from Sunil. The leaf area or leaf thickness will increase root biomass. Question 1. Question 2. Soil nutrients will enhance the root biomass in aerobic rice cultivars.
Yeah. The answer is yes. We just now discussed about the leaf and then the root when Satyendra asked that question.
Yes.
And I think the second one is when the nutrients are there. For example, it is something very, very well worked out. If there is less water in the soil, the plant tends to grow roots more. There is something which is called as a responsive trait. Root per se is a quantitative trait, but root growth rate under stress is a responsive trait. And I think it was tuberosa student called Collins, I think that paper was there in Plant Physiology in 2017. You should read this. And there they very clearly say that the moment there is stress in the soil, and the stress could be nutrient stress or water stress, the plant thinks that there is more resource available somewhere, and it tries to grow. And there is a lot of genetic variability for the response itself. This is what I call it as the propensity to respond. The moment something is under stress, the plants will grow, and the amount of biomass that it can put in terms of growing deeper roots under stress condition is a variable thing. Now, why do they vary? It is mainly because of the fact that there are mechanisms that does not make the plant or genotype to respond. Now, therefore, for sure, when there is nutrition stress or even when there is water stress, the roots will become slender and longer, thinking that there could be more somewhere available. And if they don't, they suffer. But this growth of the root is always at the cost of the shoot biomass. So again, like Dr. Rao was mentioning some time ago, it is the soil that determines the plant.
Shall I move on? He's also asking, out of the various methods of cultivation like SRI, direct seeded and alternate wetting and drying, which method is most preferable method for rice cultivation? And is the cultivation of the variety will be depends on type of soil?
Yes. I mean, which variety is better? I don't know if I can answer that at all, because it's mostly an agronomy person should be able to answer it. But for me, if I were to now compare all of them, any method which saves maximum water without any yield loss is the best method. Now, if you say semi-irrigated aerobic condition where I surface irrigate once in 10 days, developing a variety for that ecosystem is most important. That is what I would argue for that. But I do not know if you say the Sri method or direct wetting, dry direct seeded or wet direct seeded or alternate wetting and drying, which one is the better method? Wow. I'm not sure. All of them are good methods.
Sir, unmute, sir. Sundaram, sir, please unmute.
Session, when Satyendra raised the topic of role of miRNAs, and then when you mentioned that acquired stress tolerance, I'm tended to think whether is there any role for epigenetic factors in addition to genetic factors also with regard to tolerance rate?
Excellent. That's it. I mean, that is what we have to do now. In the sense, see, we have that method. For example, if you prime the seed, let us say a seedling in some kind of a gradual stress or an induction stress, and then collect the seeds. Repeatedly, you do that. I'm very sure there are epigenetic signatures.
No, like any such studies have already gone.
No. I mean, in one of our colleagues, Dr. Natraj started it, but then that did not go well for some reason. I think we should definitely do that kind of work.
We have we have an authority. You may be knowing him. He's also student of Dr. Uday Kumar. Dr. Girisha is here. Dr. T.M. Girisha Mohan.
Girish, of course, I know him.
Like we have some collaborations with him related to.
He's in some other university institution.
No, he's here in Hyderabad in Birla Institute of Technology and Sciences, Hyderabad campus.
Yeah.
I think when you are here, we'll take half a day time. We'll go and have a discussion with him, and then we'll see what's the role of epigenetic factors. He has got the tools and techniques. We just need to provide him the materials. I think we'll be investigating. Dr. Satyendra is also closely working with him, and then we can see.
Absolutely. Epigenetics, along with all your new things such as, see, there is one more idea that is coming up everywhere is the elicitors, small molecules. Which small molecules can I develop? I mean, that is the reason why I actually had a chat with the chemists, these organic chemists, and then the other agricultural chemists they call, and soil science. I don't know, soil science wouldn't do this much of in terms of the plant. The chemist, in terms of looking at, let us say you do the metabolome and you find a lot of small molecules come there. I mean, ABA is a small molecule, glucose is a small molecule, they're all small molecules. When you look at these small molecules as elicitors, and that is when you now apply them or upregulate them either by genetic engineering or by breeding or physically spraying them, agronomic, like management, there should be an increase in the response of that particular thing. So that discovery is another way that we can go forward.
As Dr. Sanjeev is our biochemist, I think
Yeah.
this is a food for thought for you, Dr. Sanjeev. You can think about
Yes, sir. Yes, sir.
Yeah.
Definitely.
possible line of direction.
Epigenetics, this microRNAs, non-coding, long non-coding RNAs.
And even a simple thing like methylation may matter a lot.
Yeah.
We can go ahead.
Emman Arun is asking, is it true that compact panicles are more often observed in drought-tolerant varieties of rice?
Oh, I'm not sure. I'm not too sure about the compact panicles. Are they drought tolerant? I'm not very sure. I mean, why would that be? I don't know. I'm not that very sure at all. I'm sorry.
Dr. Kalyani, you want to interact?
No, sir. All right, sir.
Okay. And Dr. Gurraj Katti, retired scientist of our institute, is asking, are there any physiological stress indicators common to both biotic and abiotic stresses?
Common what? Common physiology.
Yeah. Are there any physiological stress indicators common to both biotic and abiotic stresses?
Yep. One of the most common thing that happens in any stress is what we classify them as oxidative damages. There is a burst of free radical generation. If there is an insect, if there is a herbivory, if there is a pathogen, if there is drought, salinity, heat, anything, the death comes because of reactive oxygen species. So that is something, that's the reason why when we studied all the acquired tolerance mechanisms, we looked at oxygen radical generation and then management. Managing oxidative damage is most important. And there was a paper in Science several years ago that if you can manage reactive oxygen species, you will never grow old, you will die, but you will never grow old. That in human, in every animal, aging is associated with reactive oxygen species, and the metabolism becomes weaker and it produces this reactive oxygen species. They damage the membrane, blah, blah, blah. All the cascade takes place. And that seems to be one of the most common ways by which a plant exhibits its response to any stress. Aging, development, responding to any sort of biotic or abiotic stress, all things happen with that. Therefore, if you can address oxidative stress management to a very large extent, you can, I mean, that is when Sundaram was asking about we need to also look at pathogen and insect physiology kind of things. One of the things I would be tempted to observe will be differences in these mechanisms of reactive oxygen species.
That is something wherein I would like to bat for. I've been discussing with my esteemed retired colleague, Dr. Katti, and also with the head pathology. While we all know about what the insect does or what the pathogen does and what the resistance gene, what it does, but we are at a very preliminary phase in terms of cause and effect kind of relationship. What is in a susceptible variety, what are all the physiological changes happens and what it is undone in a resistant variety that we don't have the depth of knowledge.
Correct. That's what I'm saying.
Several genes.
Yep.
We know how they work, but we don't have 100% understanding about the various physiological mechanisms associated either with tolerance or susceptibility, particularly against biotic stresses.
Sure. Absolutely. That is something which is most important. That is what I was telling to Dr. Padmini and Ramesh, physiologists should have gone and suggested to the pathologist, look here, we can do this, can we work with you? And we don't do that. Isn't it?
May I now invite Dr. Subramanian sir, head physiology, IIRR, to interact with the speaker. Subramanian sir?
Sir, are you there, sir?
Sir, I invite Dr. Raghuveer Rao sir to interact with the speaker. Raghuveer sir.
Yeah, Sanjeev.
Yes, sir. Please interact.
Yeah, sir, it was very nice, interesting listening to Dr. Shesai, sir. And we are happy to be associated with you for another 3 years, sir. We look forward to you in RSC meetings.
We'll have for more years, sir. We will not allow.
Exactly. Why only 3 years?
We look forward to you, sir, as long as we are here.
I mean, I must apologize. Subara was also telling that they started discussing with me several years ago. Why it did not go up? I'm really upset. We should do this. Yeah, I mean, why only 3 years until I die? Until
that's wonderful, session. Huh?
Why do you use that word?
Yeah, sorry. Correct. We should not use such things. Whatever is obvious.
Decided by somebody else.
I know.
Enjoy till we are here.
Until that somebody else decides, we are going to definitely work together is something that I'm saying.
Yes. That's wonderful. I think Dr. Sengutuel is working on aerobic rice and then water stress adaptation traits related to hybrid rice.
Yeah.
Dr. Sengu, ask your questions.
Thank you, sir. Sir, really very nice presentation. We really enjoyed your insights on the water use efficiency. So my question is, when we do this drought, there are always we have heat stress confounding effects, especially what we say the transpiration cooling or those things. How to overcome that?
That is what I was telling a while ago. There are some of these combinatorial stress tolerance we need to understand. We all know that the moment you have drought stress, the heat will increase, and if there is a high temperature increase in air, that will lead to a very similar drought stress situations. They have to be understood because, for example, one of the mechanisms of drought heat tolerance is to cool itself by transpiration. So the moment you transpire more, the water use efficiency must decrease. This is where the problem will be. Now, when Ramesh asked that question, what other traits that we need to measure above ground? There's something called as the epicuticular waxes. What is important is that when the temperatures are increasing around the air, which is what is expected to happen when the global warming occurs. So the climate resilient agriculture, your MICRA and all those things are already addressing such things, I'm sure. So what we need to do is to reduce the heat load onto the leaf. Heat load you cannot reduce by decreasing the sunlight energy, but you can reflect a portion of that so that you don't have heat loaded onto the canopy. And that can be done by selectively accumulating cuticular waxes. And there's a lot of work that is being done. And we have also done something. One of our Preeti, as I said, her paper is under review in agronomy and crop science just now. And what we do is, and Pratibha, of course, did something excellent during her master's program. We measured the epicuticular wax content on the leaf surface and then measure the temperature of it, leaf. So wherever the wax is high and it reflects more of the infrared radiations, the heat load onto the leaf decreases. When that happens, the plant tends to keep the stomata open, but without transpiring. It is a question of what is called the vapor pressure deficit driven transpiration. This is more of a biophysics kind of a thing in physiology or in transpiration, but that can be biologically controlled. So one of the ways that we can actually make the heat and drought tolerance is to have heat load reduction by not allowing the heat to increase. And there is another metabolic way of increasing the transpiration to cool the canopy. So both are important, and therefore there will be some kind of a contradictory outputs that come when you talk about high temperature, and then drought. We need to work out on those things. But that's a good thing to now look at. But there are mechanisms. People are already working on that.
Thank you, sir. So if you don't mind, one more question. So when we see the root, because when drought tolerant or sensitive, so because normally we impose drought, it is maybe a moderate drought or a severe. So it normally it lasts for 1 month. So most of the drought tolerant has a more what is the underground traits. What I mean is, will it use more energy for a conversion of that source for the root traits? Will it affect the yield?
Yes, of course. For example, it will be something like if there is a plant which now has to invest biomass in terms of growing more root, it will be always at the cost of yield. I mean, therefore, there is something which we call as the trade-off. When you increase the drought tolerance, yield decreases. When you increase protein content, yield decreases. There is always this trade-off. We need to, it's something like your efficiency of the plant to make the whole organic matter is X, and the X has to be shared between 3-4 activities. It should be a fraction of X. That's what I'm saying. When you look at the productivity per se, we have two situations. Under resource sufficient conditions, you have to increase the potential yield. And when there is resource limitation, you have to increase the realized yield. So therefore, there are mechanisms where it becomes what you call as confounding to one another, and therefore there is going to be a trade-off. The moment the plant puts more biomass into the root, shoot will have to decrease. If you don't want that, the shoot's ability to make more photosynthesis should be done. Now, how? Look for germplasm. That's what I said. We look at germplasm, identify the contrast, and in the contrast, look at all the mechanisms in detail. But that is basic science. But the basic science has to be translatable or translational. Then that has to happen this way. That is, you screen for the variability for a trait that you think is important, where the whole biomass production efficiency is high, and such genotypes will put more biomass to the root, but the yield will not decrease. Such things exist in nature. You need to find it out.
Okay. So in that case, shall we, for example, when we see the drought tolerant identification, it should be in normal condition also in drought. Both places it should be high biomass. What the root biomass should be there, or only once you impose the drought, then it should have the more root.
Correct. That is exactly what the second sentence is more appropriate, because I don't want to unnecessarily waste my energy when I don't have any need for it. And I should be quickly able to put that. For example, there is the people talk about what is called a sensitive stomata, BPD sensitivity to the stomata. These are all two different things. If there is there is some data which I didn't show you, but there is some when you do this real through high throughput or real-time measurement of transpiration, there are plants which starts transpiration very early in the morning, and somebody who takes little slowly. Now, if you start early in the morning, your day will be longer, but your productivity will be better. This is what we have seen. But they will lose more water. Should they be allowed to do that or not? It all see. I'm not saying that your selection should be trait based, but end must be yield based. If whatever we do in molecular biology to physiology to pathology to anything, you have to ensure that the yield is high. Therefore, we must ensure that the genotype that we are finding or developing must relate to yield in whichever way we want. Therefore, the question that you asked is, if the genotypes that we are that are growing faster or growing differently, and they are responsive things. If there is stress, they should be able to put up more things quickly, and that is what we call the propensity to respond. Roots are there, but root responds to stress. Do we have variability for that? If yes, what causes that variations? And what are the biochemistry, physiology, molecular biology of that? And once you understand, then translation through breeding can be definitely possible.
Thank you very much, sir.
And now I invite Dr. Mahendra Kumar sir to interact with the speaker.
Sir, very exhaustive and very nice lecture, sir. I want only small clarification. Above ground portion we can easily assess any changes.
but root is very difficult task. Even under field conditions, it is very difficult to get the total root volume also. It is very difficult. Is there any easy way of doing things other than when you go for a field conditions, not in root rhizotrons and other things?
That's what we discussed some time back, sir. What I would say is, if the root is in the field, root measurement is definitely difficult. Because, I mean, definitely difficult, or it will be very highly erroneous. The reason is, if the moment you pull it out, you will lose half of it. So you do not know which portion, how much portion you have lost, and therefore it's not correct. But in the field, if you want to do this, one of the things that we can do is to screen leaf temperatures, which is what we did in one of our large experiments where Pratibha had developed 10,000 BC3 F2 lines. We only measured the temperature of it, which reduced the number down to 1,000. In the 1,000, we did an intensive activity of measuring the root actually. But if you have larger number, some scans, you have these days infrared cameras. You just send them across, or even put it on a drone, go capture the temperature, all of them. And they do those kind of things, which will be also necessary for heat tolerance. Temperature of the canopy is a sort of a good surrogate for root traits. Deeper the root, cooler will be the canopy, and vice versa. Because they access water and throw it out, cool themselves. That's how it is. But not necessarily. But one step towards the truth. And then take some contrast, check if they are really that way in the in a controlled condition, and then go ahead.
Sir, with this we came to the end of questions in the chat box, and now I invite our director IIRR for his closing remarks.
I think I've been interacting during the presentation and also in the question and answer session. I think we can go to the next part of the agenda.
Yes, the next part is virtual felicitation of the speaker.
Yeah. Sir, it's customary now, we are in a sort of a semi-lockdown. So it's our proud privilege. Next time when you are here, preferably during our annual RISE workshop, this is the trophy or a memento we would like to share with you.
Wow.
Where your photograph and your name, and then our appreciation is engraved. And congratulations, and then we are highly thankful to you for delivering today's talk, sir. And I hope all these interaction and then discussions will continue further also. Thank you very much, sir.
Of course. Thank you very, very much.
Thank you, sir.
I was wondering now. Wow, I tease all these my friends who call me for all these talks. Sundaram. I said, if I had come there, you would have given me some whatever chocolate. But now you are showing. My God, I didn't expect this. I mean,
we'll show Hyderabadi hospitality once you come here.
Thank you so much, and I really, really am hoping I will come as soon as I can.
Yeah. Certainly, mark your dates in the second week of preferably around second Saturday, Sunday time. We'll be having our annual RISE workshop.
Yeah.
We have we'll consult our colleagues at NRRA, and then we'll get permission from ICAR and then get back to you.
Sure, definitely.
Sir, now I invite Dr. R. Mahendra Kumar sir to propose formal vote of thanks.
Yeah, and before that formality, I'll also thank all of you. And so many of you overwhelmingly sort of listening. This actually has gone to more than 2 hours, right? Almost.
Yes, sir. Yes, sir.
So
there were more than 120 participants at a given point of time. When you finished your talk, there were more than 120 participants. And in YouTube also, I was seeing say almost 300 people were watching the.
Oh, it's on the YouTube as well.
Yeah.
Wow.
So you cannot escape from us. You're always already there.
I will not even try. I mean, one of my it had been always a dream that how is it possible that I can work with all of you in the RISE workshop and RISE meeting and the RISE group. It has always been elusive, and I'm very, very, very thankful for all this. And thank you so much, Sundaram and Padmini Didi. It's been a wonderful time. I used to call her Padmini Didi. I do not know if I can tell that. When you call me Shesha, I think I can also do that. So I think we are already a small family of people, and I'm sure we can do that. And thank you so much for giving me this opportunity.
Thank you.
Thank you, Dr. Sanjeev. So I'm very glad to propose a formal vote of thanks. Sir, I think it was one of the best, I think, webinar I had for last. We are having more than 10 to 12 webinars, and it was one of the wonderful webinars, and it was very informative. And the way you responded for all the queries without irrespective of the subject and all other things, it was really excellent, sir. And on behalf of the Society for Advancement of Rice Research and also IIRR and also NRRI, we sincerely thank you, sir, for your valuable time and your knowledge sharing. Thank you very much, sir, for your this wonderful session. I would like to thank our Dr. Padmini Swain Madam, Director NRRI, for I think involving us also in this Azadi Ka Amrut Mahotsav series of webinars, and especially our Director Dr. R.M. Sundaram, who is very dynamic and is very encouraging for our scientific community at IIRR, and is also very close to you also, sir. So thank you, Sundaramji, for making it this happen, this webinar happen. And special thanks to our speaker. And I think I did not see anybody who can explain everything very without any hesitation, sir. Thank you very much, sir, for your wonderful speech and also your clarifications. I would like to thank ARRW members and also SAR members who are attending across the country. And also I have seen some of our seniors, Gurraj Katti and many others, who are also still they are interested in this subject. I thank you them. And I also seen that PJTSU Thati Ramesh, physiologist, in this virtual webinar. I would like to thank all the heads of the sections of this IIRR and all the scientists. And it's a very good participation, sir. I was really surprised. It was more than 150 participants are there at one time. I would like to thank. I think that knowledge will be very useful for all the people who can work on drought. Now it is water is going to be very critical in coming years, sir. Your work will be very much appreciated in coming years also. I would like to thank our especially Dr. Shailaja, who has made all the webinar arrangements, and also the team of our webinar team, that Dr. Padma Kumari and Dr. Sanjeev, and all other people who are directly or indirectly involved in this arrangements. Thank you one and all, and it was a great learning experience for all of us. Thank you, sir.
Thank you.
We'll meet you when we come to Bangalore, sir. Thank you very much.
Absolutely, absolutely, sir.
Even by 1 minute, invite him for Hyderabad, sir, or workshop.
Yes, sir. I will definitely come. Thank you,
sir. 1 minute, sir. I request all of you to switch on your videos, and I request Gayatri to take a photo.
Okay, sir.
Because this will appear in ICAR's website also.
Please,
Every time.
please, whoever is there, please switch on your videos. Gayatri, you take, you go to the other screens also. Screen 1, screen 2, and screen 3.
Okay, sir.
You have to tell us how long we keep smiling.
Please, sir, you should have a virtual smile, sir.
I think you indicate when it is done.
Yeah, or somebody tell a joke and we keep on smiling. There is a door, it seems, Sundaram.
Yes,
if you only smile in front of the door opens. Otherwise, it will not open at all.
It's true to our life also, sir. When you keep smiling, all doors will open.
Yeah, simple.
Done, sir.
Thank you.
Thank you,
sir.
Thank you.
Now
Thank you all, sir. Thank you all.
Thank you, sir. Thank you. Namaste, sir.
Namaste, sir.
Thank you very much, sir. Thank you, Dr. Patra. Thank you very much.
Thank you very much.
We'll shortly get in touch with you and then decide the date for the workshop.
Thank you, sir. Thank you.
Thank you. So the meeting is closed. We'll keep interacting session.
Yes, sir. Thank you very much, Sundaram. Papa.